Hello and welcome to another episode on the microbial world. Today's learning will be on propagation and maintenance of industrial microorganisms. Introduction. Microorganisms form the biological backbone of industrial processes, serving as tiny factories for producing everything from life-saving antibiotics and enzymes to biofuels and bioplastics. The industrial value of a microbial strain, however, rests entirely on the ability to maintain it in a viable, pure, and highly productive state over long periods. Objectives of propagation and maintenance. The primary goals of a culture management system are to obtain an exotic, pure, and physiologically active culture for continuous industrial use, preserve the genetic and metabolic stability of the production strain, preventing desirable traits from being lost or reduced, ensure consistent product yield and quality during repeated fermentation batches. Sources of industrial microorganisms. Industrial strains can originate from several sources: natural isolates, mutant strains, genetically engineered strains. Culture collections, authenticated, certified, and preserved microbial strains from organizations like ATCC American Type Culture Collection, providing a reliable, pure starting point. Propagation of industrial microorganisms. Propagation is the careful, staged process of growing microbial cells from a preserved state up to the massive volume required for the final industrial bioreactor fermenter. Stages of propagation. Inoculum development scale-up. The goal is to introduce a large number of healthy, physiologically active cells into the final production medium. This is achieved in a multi-stage scale-up process. Stage 1. Stock culture, the blueprint. Stage 2. Seed culture intermediate. Inoculum development, the scale-up. The culture is transferred sequentially from smaller to progressively larger vessels, for example 1L to 50L to 500L seed and industrial fermenter. The size of the inoculum for each stage is typically 5 to 10% of the new working volume to ensure rapid entry into the productive log phase. 4 point-to conditions for successful propagation. To ensure high cell viability and metabolic activity. Parameter optimal condition. Importance. Temperature. Should match the organism's growth optimum for pH. Maintained near neutral for bacteria. Slightly acidic for fungi, around 4.5 to 6.0. Aeration. Aerobic microbes require sufficient oxygen. Anaerobes must be protected from air. Nutrients. Must support both growth and metabolite production. Maintenance. Preservation of industrial microorganisms. Maintenance involves keeping microorganisms alive, uncontaminated, and genetically stable over time without losing their desired traits, because frequent subculturing can lead to mutation and strain degeneration. Proper preservation methods are essential. Types of maintenance techniques. Short-term maintenance, active culture preservation, used for regular laboratory work or frequent inoculum preparation. 1. Periodic subculturing. The culture is grown on agar slants. Storage under mineral oil, paraffin oil overlay. Agar slants are covered with sterile mineral oil to prevent drying and oxidation. Long-term maintenance, stable preservation. These methods maintain cultures for several years without subculturing. 1. Cryopreservation, freezing. 2. Lyophilization, freeze drying. Silica gel preservation. Drying microbial suspensions on sterile silica beads and storing in sealed vials. Commonly used for Streptomyces and Bacillus species. Liquid nitrogen storage. Cultures stored in liquid nitrogen at -196 degrees Celsius remain dormant and stable for decades. Ideal for long-term preservation of valuable industrial strains. Avoiding strain degeneration. Strain degeneration is the loss of the desirable industrial trait, for example reduced antibiotic yield due to mutation or contamination. The seedlot system mastered working culture. This is the mandatory preventive measure. The master cell bank MCB is kept safely preserved and is rarely used. Avoid excessive transfer. Limit the generation number transfer cycles between the MCB and the production fermenter. Maintain records. Rigorous documentation of strain lineage and performance history. Quality control in culture maintenance. Quality control ensures that the culture is pure, viable, and still capable of high productivity. Quality check. Purpose, method, purity testing. To ensure the culture is contaminants-free. Microscopy. Gram staining, isolating on selective, different media. Viability testing. To ensure a high percentage of cells are alive after preservation and revival. Colony forming unit CFU counts or dye exclusion tests. Productivity check. To confirm the strain still produces the target compound at the expected yield. Assays to measure enzyme activity or antibiotic titer. Genetic stability tests. Scale-up and industrial inoculum preparation. The final step is preparing the production inoculum. The key objective here is to have cells that are pure, validated by quality control, physiologically ready, typically transferred during the late log phase, when cells are growing rapidly and are metabolically most active, ensuring a quick and robust start to the main fermentation batch. Safety, record keeping, and strain documentation. Standard operating procedures SOPs, detailed, mandatory instructions for every step of handling, transfer, and disposal to ensure reproducibility and compliance. Biosafety, documentation, all preservation dates,